Hi everybody, welcome to Unit 3 Biology. Today we're talking about how we get from DNA to proteins. So we're talking about the steps of transcription, RNA processing and translation. Our study design dot point reads the genetic code as a universal triplet code that is degenerate, and the steps in gene expression including transcription, RNA processing in eukaryotic cells, and translation by ribosomes. So there's a few key words that we need to know for today, and there's also some very important processes. We do have 3 review questions based on our last video about DNA and RNA, so you can pause the video here to give those questions a try. Here we have the answers to those questions. Check your answers against these. Please remember that as long as you've hit the important information, the wording is allowed to be different. You do not need to word your answers like a model answer in order to get the points or the marks on your exam. So, we do need to start off by briefly talking about proteins. Proteins will come up again later in the study design, but you need this little bit of knowledge for today's lesson to make sense. So you would have heard of protein before. You know that people who go to the gym might use protein powder. You would have heard that a lot of meat products would have protein in them, and even some plant products have proteins. And that's because they're essential for life, so they're really found everywhere. Proteins can be structural, they can be used to build. They can be enzymes, so they can be used to digest, or also to build, in fact, but we do talk about this later. Some hormones are proteins, so they can be used as signaling molecules or messages. Some proteins are regulatory proteins, which means that they tell the genes what to do, or when to do it rather. There's lots more examples. You don't actually need to know about these for the study design, but it's a good bit of background knowledge. The building block of proteins are amino acids. So to actually build a large protein structure, you need to have a long chain of amino acids. And again, we talk about this later in the study design. A key piece of information is that there's only 20 different amino acids. Humans can make some and need to eat the others. The amino acids that can be made by a living thing tend to vary depending on what that living thing is. So we're not going to go through and name them or anything because it does vary so much. But just so you know, some we can make, some we must eat or ingest to have access to it. But remember, there's only 20 different ones. So think about all of the different types of proteins there are. That comes from just having 20 different amino acids in different combinations. Which brings us to our genetic code. This is the study design dot point. It says it's universal and triplet. So that universal means that DNA is the same no matter what living thing you're looking at. So that means that the DNA from a bacteria could be picked up and put into a human, and it would be exactly the same. Wouldn't necessarily work if we just did it like that. There's other steps involved. But the DNA that's in you is the same as in me, is the same as grass or cats or snails. That DNA is all the same. We say that it's a triplet code because it's read in groups of 3. And that's really important for today's work. So when we've got our DNA and we're reading it, we look at a group of 3, the next group of 3, the next group of 3. And this is really important. We say that the genetic code is degenerate. So if we do some maths, we know that DNA is read in groups of 3. You've got 4 combinations for the first position, A, T, C, or G. You've got 4 combinations or options for the second position, A, T, C, or G. 4 options for that third position, which means all up you have 64 possible combinations. We won't list them all here, but just think you could have AAA, ATA, AGA, ACA, and so on and so forth. 64 of those combinations. But we know there's only 20 amino acids. So what that means is that you can actually have multiple triplets, multiple different combinations of DNA triplets that code for one amino acid. And you'll see this really soon. There's a fantastic table that we've got that we'll look at. And you'll see that sometimes there's a whole box for just one amino acid. And that's because there's multiple combinations that lead to it. We'll come back to this one when the table is up. So as an overview of these steps, we see that DNA in the nucleus is read and transcribed into pre-mRNA. So that's step 1. That's our transcription. Step 2 is that the pre-mRNA is modified and processed into mRNA, which then leaves the nucleus. And step 3, that mRNA travels to a ribosome and it's read or translated 1 triplet at a time. So those groups of 3 are read in order. As this is happening, our tRNA is bringing the correct or the appropriate amino acid to the ribosome to help build this protein. Now you might have noticed here, I'm using language like nucleus, which tells you that this is a eukaryotic cell. This is a very similar process in prokaryotes, but we do have a little bit less to know. So in prokaryotes, the DNA is read and mRNA is formed. So we have a modified step 1 with less steps in it. And then we go straight to step 3. So we skip step 2. We have a new step 2. The mRNA is read by a ribosome. So you need to know that this step here, the processing of mRNA, that doesn't happen in prokaryotes, only in eukaryotes. If you're ever asked a question about prokaryotic transcription and translation, it is a much more simplified process. Now we are going to go into detail for each of these steps. We're going to assume that we're talking about eukaryotes, but again, as long as you ignore the processing stage and the language of nucleus, it's exactly the same for our prokaryotes. Now, transcription is really important in eukaryotes because DNA is actually a very large molecule. It's so large that it can't leave the nucleus. We know that our nucleus does have pores in it, so it'll have sort of just little gaps. But our big pieces of DNA, they can't get out those pores. So what happens is that in transcription, we create a copy of just the relevant gene. We don't want that whole chromosome. We just want that little section that's needed right now. You don't need to know how the system or the organism knows that that part is needed. That's way beyond the scope of VCE biology. Just know that there is a way that the body, the organism, the system knows, hey, I need this particular protein. I'm just going to go and transcribe this gene. By transcribing just that one gene, that one section of the chromosome, we make pre-mRNA, which will lead to mRNA. It's a much smaller molecule, and so it can actually leave the nucleus via those pores. It's also only a temporary molecule. Once it's used, it gets broken down so that all of those parts can be used again later on. So what we have, we've got our long strand of DNA. It's all wound up in that double helix. And what happens is just one section, the section with that gene, it unwinds. So just that one section becomes straight and it unzips. So that means that this bit here where the bases have a bond, they sort of separate out, they leave a gap. So we could draw it, we've got our helix and then it straightens out and a helix. Like this. And our bases now have that gap in between, so they're not directly bonded. Unwind, unzip. We have this special enzyme called RNA polymerase. We will talk more about enzymes down the track, but what you need to know about this one, polymer is something that's made up of lots of parts. So polymerase is something that builds. And in this case, it's RNA polymerase. So this enzyme is building RNA. The enzyme reads the DNA. So it sort of latches on and it's reading this code. And what's happening here is that as it's reading, if we have A, T, C, A, G, G, T, it's building a strand of complementary mRNA. So complementary to the A, well, it's normally T, but in RNA, we only have U's. So we'd have a U. Our DNA is a T, so complementary, that would be A. C would go with G. A again would be a U. G goes with C. G goes with C. T goes with A. So we're building our pre-mRNA that is complementary to that strand of DNA. Now this terminology doesn't tend to come up in VCAR exams, but does tend to pop up in practice exams, so I'll briefly talk about it. We have a coding strand and a template strand. So what happens is that the RNA polymerase is reading the template strand. And think about it just like if you were going to make a cake in the shape of a teddy bear, you would use a template for that. So we read from the template strands. Our mRNA, because it's complementary to the template strand, is then pretty much the same as that coding strand there. If we have a quick look and build it, A would be T, T would be A, C is G, T, C, C, A. So you can see that coding strands and the mRNA pretty much the same, just swap out your T's for U's. Again, this doesn't tend to come up on VCAR exams. Can't say that I've seen it often. It's a good little piece of knowledge to have that'll separate you out from the rest of the pack. But don't worry if it's a bit confusing, because at most that'll be 1 mark on an exam. It's not the be all to end all. So again, another example, here's our coding strand. We're going to fill in our template strands first. So this is all a long, big strand of DNA. We have A goes with T, C goes with G, and G goes with C. This is a really great one for you to practice. It's not uncommon for VCAR to ask you to write a complementary strand of either DNA or RNA, so make sure you practice both and that you know which pairs go together. Okay, so we've just built our template strand where you can see that they're all complementary to the coding strand. Now in my green pen, I'm going to go through and make the RNA, or the pre-mRNA. So again, we're reading the template strands. Okay, T normally binds with A, it's the same in mRNA. Ooh, now A would normally bind with T, but RNA doesn't have T, so it has to be a U. G with C, G, A, and so on. I'll just quickly fill these in. If this is one that you feel like you need a bit of practice on, rewind the video, pause it, and see if you can make them yourself. So now we have our pre-mRNA. Really great thing to practice. So once you've built your pre-mRNA, we need to process it. And remember, this only happens in eukaryotic cells. This does not happen in prokaryotes. Bacteria do not need to process their mRNA. So this is just a matter of making that mRNA strand more stable, more versatile. So in terms of stability, processing adds some structures to it to make sure that it won't fall to pieces before it gets to its next step. And the versatility, we can actually modify or process our mRNA in different ways to code for slightly different proteins. So this step is pretty important. So there are 3 key things about mRNA processing. At our 5' end, we have the addition of a methyl cap. So it's just like a little alcohol group that goes on the end to sort of lock it in and make sure it won't break. At our 3' end, we have the addition of a poly-A tail. So it just adds a whole lot of adenines to the end there, again to make sure that it won't break. Think of it a little bit like the aglets on your shoelaces, those little plastic caps that prevent your shoelaces from fraying. And in eukaryotes, in processing, we also have splicing, the removal of introns. So what that means, if we were to draw our little pre-mRNA strand here, we can actually cut this up in different ways. So we have sections called exons and sections called introns. And these will vary depending on what protein we're trying to make. But basically, the enzymes will go along and it will chop out some different parts of that pre-mRNA, some different introns. And we say they're introns because they end up staying in the nucleus. If we color those exons maybe in orange, so this section here, 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 and here, those exons will then bind together and they will be the bits that leave the nucleus. Don't forget, we add our methyl cap and a poly A tail. So again, these sections, they just stay in the nucleus. They end up being broken down back into their base nucleic acids to be reused again. So just to reiterate on splicing, if we cut out different sections, we can have a gene that codes for a variety of proteins. And let's quickly show that in a different way. We might have a gene and say it's made up of these numbered sections. Now, if you're looking to build one particular protein, you might splice out 3 and 6. So this is what your mRNA would have. Your 3 and 6 have become introns, and they are going to stay in the nucleus. This section here is what will travel out as a part of the mRNA. But we could also cut out the 2, the 4, and the 7 and make a different strands of mRNA. Or we could cut out the 1 and the 7 and again have a different strands of mRNA. You don't need to know the process of how this happens. You don't need to know how the cell or the enzymes know which sections to cut. You just need to know that it does happen. This comes up occasionally, not every year, but occasionally. There'll be a question asking how one gene can code for different proteins. So this is where you would talk about alternative splicing of introns. After this has been processed, we go through translation. So that mRNA with its methyl cap and poly A tail travels to the ribosome and it attaches there. The ribosome will read one triplet at a time, and you can see in the image there that it's sort of attaching to one triplet or one group of 3 at a time. Those groups of 3 are called codons because they code. Our tRNA molecules with the complementary or matching anticodon will bring an amino acid to the ribosome. It will always bring the same amino acid. So for example, the codon AAG will match with the anticodon UUC, and UUC will always bring the same amino acid no matter what. It doesn't change. UUC won't bring lysine one time and then arginine another. It'll always bring the same amino acid. And you can see that as the amino acids are brought, there's sort of these little balls here. They end up creating a polypeptide chain, which is the first level of protein hierarchy. It's the first stage of making a protein. Our tRNAs will then just go back off into the cell. They will end up picking up another amino acid, but they don't need to come back unless the mRNA has the matching codon again. Now, not in this video, but there are some really great videos on YouTube where it shows this process as an animation. Really great to watch to visualize what's happening. Now this is that chart I was talking about. This is called a codon chart. You need to know how to read these. It does frequently come up. So say we have an mRNA strand that reads CCU, CCA, AA, G, GAU, for example. We need to first break it into triplets. Then we look at each codon individually. CCU, okay. Our first letter is C. Our second letter is C, so we're going to look into this box here. The amino acid that's brought is pro. Okay. Our next codon is CCA. So same thing, first letter is C, second letter is C. We're in this box here. CCA is pro. And that's an example of our degeneracy. We have two different codons, two different combinations that are actually matching to the same amino acid. This comes up again in Unit 4 very briefly. If you just have a single base mutation that results in a change from that U to the A, it's actually no big deal because you end up with the same amino acid and hence the same protein. Let's finish off our strands. AAG, okay, our first letter is A. Our second letter is A, so we're in this box here. AAG goes with lys or lysine. And we've just got one to go. Of course, in real life, there's a lot more than this, but you're rarely asked to do like more than 10. 4 to 5 is probably a good amount. GAU, our first letter is G. Our second letter is A. If we have a look in this box, GAU is ASP. So these are the amino acids that are building this protein, and it's in this sequence. You do not need to memorize this chart. You do not need to memorize all 20 amino acids. A chart that looks like this or similar, sometimes there's circles with bits coming out, will always be provided to you on your exam. Now, as one last note, we can see that we have some stops. So if the ribosome reads a stop, just nothing happens. There's no amino acid brought, that's just the end, and the polypeptide chain will detach. So these three codons, they do tell the ribosome that, hey, this is the end of the protein. And you might have noticed that there's one bolded here and in a different color. AUG or MET. Well, that's the start of every single polypeptide chain. And this, again, it's what tells the ribosome that, hey, this is the start. You can't just start here at CCA. You have to have an AUG or a MET to start building this polypeptide. So this has been our brief introduction to transcription, translation, and that intermediate step of RNA processing. Make sure that you do a lot of practice around this with whatever textbook you have access to, with lots of exam questions. These questions do tend to come up every year, particularly around transcription and translation, in varying amounts of detail. It's a key part of our study design, and it's one that you would be well off knowing very well. Thank you for listening. Hope you have a great day.